Hi, my name is Patricia Melloy. Thanks to the organizers for the opportunity to present today. And I'm presenting a collaborative project that we're working on here at. So this is a project that we're doing in an intermediate level biology course. These are students who've already taken Bio 1 and Bio 2. And they're taking it at the same time as genetics lecture. So that's a large class that's split into 5 lab sections. And typically the students work in teams of 2, but we have some flexibility if there's a preference for a slightly larger group or they prefer to work alone. And some of the content that is already covered at this point in the semester when we do the independent project includes things like managing a model organism. We heard in the previous talks about creating a figure, revising it, creating a figure again. We do that as well. They've learned about data presentation, experimental design, and they've done various techniques, including things like molecular genotyping, and they have carried out a smaller project on the effect of DNA damage on yeast at this point. So we are developing what we're calling for now a mini cure type project. And the learning outcomes that we have for this is to make it completely student driven from the beginning. And so our goals include helping the students experience, confidence and independence, be able to demonstrate their ability to design an experiment, analyze results, and choose how to present the data. And then we hope that they'll be able to take the things that they've learned in genetics lecture and gain a better understanding of how that applies to things in the lab. This is meant to take 3 lab meeting periods that are about 2.5 hours long. So in the first one, they design their hypothesis, and we like to emphasize that the students are actually choosing their own independent variable to manipulate. So even if they're working on a project that comes from a kit, they're not meant to exactly follow the protocol. They're meant to create a twist on it, something that's different about this time. And then another thing that the students do is create their own material shopping list. And this helps improve communication and planning skills and working as a team on that. And then they can modify an existing protocol or create a new protocol so they plan out the experimental procedure. And I also want to point out there's several places for instructor feedback along the way. Then in the second lab period, they conduct the independent project. And before they move on to the third part, though, there are a few open lab period opportunities where they can check on the progress of their work. And then in the third lab period, they troubleshoot or collect data, analyze their results, and they begin to decide how to present their data. And at that point, we have the lab report template that we give them at that time. The lab reports due 2 weeks later represents about 20% of their grade in the lab part of the course. And I dropped the PDF of the lab packet in the chat. It's also available through microBREW, but this is just a snapshot of how we help them step through different parts of the process. So this is the section on designing and planning. And so you can see how the students think about what research question they want to ask, what variable they want to manipulate. They design a hypothesis, then they gather their materials list, and then they set out a particular experimental protocol or procedure that they have ready when they come back for the second lab period. I wanted to give you another view of how we organize this big thing, right? So one of us is an instructor and also the coordinator for all the labs. That's Miriam Marks in this case. And she's developed a Google document where the students can fill out this Google form actually and put in their materials list, and it'll automatically populate a spreadsheet for her. And we've given you a snapshot of what that looks like to the student, or the output, sorry, the output from the student. And then we also have an opportunity where they can ask questions there as well and get feedback from us on their design and materials, because some students, they may not speak up in the lab, but they like being able to ask questions in this way as well. And so in the pictures, you can see how Miriam takes this list and kind of figures out how to organize everything, lays out some materials. On the right, you can see some media that's used. But if the student wants a special kind of media, of course, we're open to that, and they can make that themselves if they give us all the ingredients in the shopping list. Just wanted to give you a example of some of the projects that they choose. So they can do something based on something we did earlier in the lab, or something a little different. So many will choose some type of genotyping. Others will do a modification of our DNA damage project working with yeast. Some will look at the effect of DNA damage on planarian regeneration. Some can look at more of a molecular style project that we did early on, looking at transformation of the P. glow plasmid in E. coli, and not just following the kit, like creating some kind of variable in the protocol. And then like to talk about the chia seeds thing as an example of how the feedback can make a difference. We have some students that really wanted to do something with plants because of their interest from a different class. But then when we talked to them, it kind of became clear that it might be challenging to do this within the time frame that we have. And so we're able to introduce the idea of, well, do you want to try something with chia seeds? We can obtain those, and perhaps the effect of DNA damage on chia seed generation. And so that seemed to work out well. We had a couple students do that this time. So just to summarize a few kind of highlights of the results, we were really pleased that a little over 72% of the students were able to obtain usable data on the first try. About 20% took some of the open lab period time to begin to plan for repeating the procedure or altering it to make it more successful. That's about 20%. Only about 6% did not have analyzable results. We also found, and we're just starting to get into assessment now, that the grade on the independent report, which is all student designed, the median ended up being quite similar to the grade that we saw for the last project before, and that was the DNA damage report. That was something that we designed. So they seem to be able to take what we did and then run with it and make it their own. And this big project can be intimidating, but we tried to introduce it very slowly and in pieces. So we were pleased that about 95% of them did submit a final report on time. So just to wrap up here, our future directions include things like trying to, and this relates to actually what our keynote speaker brought up, Sarah brought up the idea of error framing. So how can we emphasize the value of collecting all data, including failed experiments, and then troubleshooting? We're playing around with the design of the open lab period. We would like to improve the students' skills in terms of how they learn about how to present their data, because this seemed to be a slightly weak point when we make it very open-ended and they have to choose how to represent the data. Potentially, we want to add self-assessment or a survey about how confident they feel before and after, perhaps some more pre and post-lab assessment of skills. So thanks to everyone who helped with this project, and we're happy to take questions. Thank you.
Great, thank you for the talk. So does anyone have any questions for Dr. Melloy?
And please call me Tricia too.
I was just wondering, did all the students use the same procedure for introducing DNA damage? Is that what they were doing in this?
So I didn't talk about that, but they were using a UV light box to affect some yeast that were either wild type or UV sensitive in the previous project. So there were some who chose to kind of riff off of that. Others, they did have access to ethidium bromide in a very controlled way. They could use that as a way to try to affect the DNA. But most of them, they were using one of those approaches.
Joseph, go ahead.
Hi, thanks for the talk. I'm curious about student choice of projects and
Mhm.
whether or not that's assessed, which I've never really thought about before. I've had this issue as well, where I teach genetics cure as well, and giving students free reign, they might choose something that's not actually genetics related. I'm curious if
Yeah.
you have those experiences and how you manage that, especially if assessment is part of that. Do students get
Mhm.
graded on the quality of the idea that they come up with, or how do you manage that?
Can I take this one?
Right. Oh, go ahead. So my collaborators here, and we'll talk about feedback, I'm sure.
Hi, Miriam. So that like one of the first things that also that like Google Forms spreadsheet helps with is that I can flag also for the other instructors when I don't know if anyone ever I had this like this pet peeve of mine personally of this like middle school science fair project of like mashing up strawberries and isolating their DNA using dish soap. And so like there are times where students like Google genetics experiment and then like they propose something where it's like that's not actually going to work. You can't just like put soap and strawberries into a PCR machine and expect like results. So there are some students who struggle with understanding protocol and like why it works and how it works. Like there are different levels of ability. And that also is tied to how the genetics lecture is mastery based learning. You cannot move on to a higher level until you get mastery on one of the earlier concepts. So we have to design the lab at the end of the year knowing that there are different levels of skills. There are students who have pretty much gotten an A already and finished genetics, and there are some people still stuck on mitosis and meiosis. So I don't think it's like a fair practice to grade students on the quality of their proposal, especially because it's their first time ever possibly conducting their own research. And instead, it's more like oh, hey, Trisha, your students have no idea what's going on. And that was actually, I think, the group that I was like, how chia seeds?
Right.
And stuff like that, where it's like, I want people to have the process of like experimental design and like inherently there's failure in that. But when you can see that like there's a clear disconnect of understanding what is the appropriate way to assess the question that they're asking. Like some students just needed more guidance and conversation early on because they just like got overwhelmed with options. And the other thing is if you check out the packet that Trisha dropped in the chat, the first page gives them context as well as hyperlinked recommendations of things where it's like we know it will fit in the time frame and we know that it is relevant. But absolutely, every time I've done this, I've had way off topic, like that's not genetics or that's not how science actually works. You know, like it, and but that's just part of the reason why we do it in the first place. But we don't include that as an expectation. It's just send me your proposal so we can make sure you feel confident on that first day that you start experimentation.
Mike, you had your hand raised.
Yeah, so just as a follow-up, I'm wondering if you leverage that opportunity to talk about testable and untestable questions. So, and maybe that relates to Joseph's point or question or inquiry about, at that point, it doesn't really matter the level if students are, still grappling with cell division or looking about molecular genetics, but there are testable and untestable questions.
Yeah.
And maybe that is something that you could assess and really be deliberate about. Or
Right.
maybe you do it already. I don't know.
Do you want to take that one as well, Miriam?
That's up to you, but it's like That's again, a lot of students are successful in designing testable questions, in part, I think, because that packet, the inquiry packet, that we shared, and you guys are welcome to use for your own purposes, but students pretty quickly realize when they like start an experiment that they made a mistake. Like
Yeah.
you can talk all you want to students about testable versus untestable, but sometimes I actually choose to let them realize that the way that they design their experiment isn't going to give them the answer. So that's why we also like included that data on like how many students got it on the first try and how many students
Yeah.
wanted to put sunscreen on yeast to see if that affected the survival rate, but they didn't actually think to measure the amount of sunscreen and they just sprayed it like they would on humans. And like they would realize pretty quickly that they made mistakes, or they made not just like procedural errors, but also like I got results, but it's not answering the question I asked. And again, like it's almost like I want people to make mistakes and realize that like every experiment they've ever done in undergraduate has been heavily controlled by their instructors and play tested to be successful, but that's not what the reality of science research is like. So it's almost like if you fail, that's also in my mind like a win in a way, because they still have to communicate it. And if that means they didn't have a testable question and in their results they're being critical of early in their experiment, their design, awesome. They learned something about this process.